So just to let everyone know that we're now opening for sponsorship through Patreon. So this way we can do it openly. So either you individual or institute, non-society or industry, if you think that you'd like to sponsor us, you can log into patreon.com and then find WebEVTalk there. And for this session today, we'd like to thank Ohriba Scientific, who's been our patron for a few weeks now, a couple of months, I think. And yeah, thank you so much for supporting WebEVTalk. So it's a great day today, 29th June 2021. Brisbane is starting the lockdown for 3 days, and hopefully that stay 3 days, not longer than that. But I'll welcome very much Gregory Lavieu from France. It's an exciting session today because Greg is very well known in the field. Greg Lavieu is a cell biologist and biochemist expert in membrane trafficking. He previously identified a new mode of transport through the Golgi apparatus dedicated to large cargoes that cannot fit into classical transport vehicle. He's currently focusing on EV delivery and aims at identifying the core machinery governing this process. So today, his lecture is going to be
Good morning or good evening, everybody. So it's my pleasure to talk today about our recent results on EV delivery on multiple time zones. And thank you, Carolina, for the invitation. So today I'm going to discuss the cellular and a little bit of the molecular aspect of the extracellular vesicle uptake and content delivery. But first, I would like to acknowledge people in my lab, especially Emeline Bonsergent, a PhD student, who did most of the work that I'm going to present now. And I would like also to acknowledge my collaborator and the funding agency. So I will only provide a very brief introduction because I'm sure that most of you are very familiar with extracellular vesicles. So as almost everybody knows, so extracellular vesicles can be secreted and released from donor cells and deliver bioactive molecules such as nucleotides, proteins, or lipid into acceptor cells. So today we don't really know the mechanisms that are used to deliver the content of the vesicle inside the cells, and that's what we are interested to investigate in my lab. Especially the docking and the fusion process that are likely to be involved in that process. So by analogy with viruses, there are at least two pathways that can be used by extracellular vesicles to deliver the content. The first one would be the most direct pathway, which will consist in first docking at the cell surface and then release of the contents through membrane fusion between the vesicles and the plasma membrane. So the second delivery process could be more indirect and also used by many viruses such as VSVG. So first, the vesicle will get internalized inside the cells, confined in the endosome, and then release the contents through fusion with the endosomal membrane. So an easy way to distinguish between these two pathways to test if the acidification is required and sufficient to trigger the EV content release. And for many viruses, the acidification inside the endosome is a trigger to the fusion process. So to test that hypothesis, we developed a new cell-free assay to directly test our hypothesis. And all the detail can be found in the papers that have been published a year ago. So the principle of the assay is very straightforward. So what we did is we isolated extracellular vesicles that contain a cytosolic cargo that is trapped inside the vesicles. So in the buffer, we had protease, and normally if the cargo is inside, the cargo is expected to be protected from protease digestion. So we mix the extracellular vesicles that contain the cargo with plasma membrane sheets in suspension. And so then we test the content release that could occur, for instance, if there is fusion between the target plasma membrane sheet and the donor EVs. And of course, because it's an in vitro system, we can control the parameters such as the pH. So in brief, we developed a very simple protease protection assay with donor and acceptor membrane to test if pH could trigger the content delivery of extracellular vesicles. So we first generate a donor cell line that overexpress HSP70, a chaperone that is known to be present in many types of vesicles. So we pick that cytosolic cargo because it's a generic cargo present in many types of vesicles. And then we purify, we isolated the extracellular vesicles through ultracentrifugation. So we first characterized biochemically the EVs, and as expected, the EVs fraction were enriched for classical markers such as CD63. They were completely depleted of ER markers such as calnexin, and importantly, we were able to find presence of the GFP tagged HSP70 or cargo. So when we did the morphological characterization, as expected, the vesicle appears as spherical structure that were harboring approximately 100 nanometer diameters. So with that, we have the first entity for our assay, the donor EVs with the cargo. But what we wanted to make sure that the cargo was truly inside the vesicle. So for that, we did a classical protease protection assay. So here what you can see is a Western blot, and here is a GFP tag cargo HSP70. So now if you only treat the sample with Proteinase K, as expected, you don't see the degradation because the cargo is protected by the membrane. And now if you digest the membrane and with detergents such as Triton, so now you have the protease has access to the cargo and can digest the cargo, and that's quantified here. There is almost a complete degradation of the cargo in presence of Triton and Proteinase K. So that was the proof that the cargo was truly inside the vesicle and therefore appropriate to test the content delivery. So the second entity that we needed for our assay was a plasma membrane sheet in suspension. So for that, we developed a new protocol that is detailed in the paper and that I'm not going to explain here, but I just want to show that when we characterize the plasma membrane sheets, so as expected, they were positive for a chimeric plasma membrane protein that we use for the purification of the membrane. They were also contain positive for integrin, a plasma membrane protein. But importantly, the plasma membrane sheet were negative for either cytosolic markers such as actin, lysosomal markers such as LAMP1, or Golgi protein. You can note that we always found traces of ER marker, perhaps because there is abundant cortical ER that remain associated with the plasma membrane sheet. So when we characterize morphologically the plasma membrane sheet, as you can see here, so we can deposit them on glass and they appear as large spread of membrane sheets. And they are positive when they are incubated with a fluorescent lectin that recognize glycan chain exposed at the cell surface, confirming that we were dealing with a plasma membrane. And at the electron microscopy level, they look at the network of membrane. So with that, we were confident that we have the two required entities, the donor EVs and the putative acceptor membrane. So now we could perform the assay, and so we mix together the donor EV with the target plasma membrane sheet, and we treated with plasma membrane, and we trigger the acidification. So as you can see here in lane 3, when we only incubate plasma membrane sheet and EVs at pH 5, there is no content release because the cargo is still protected. There is no degradation by the Proteinase K. Okay. So as I showed before, now if we treat with Triton, that's a positive control. So again, there is full access to the cargo, and the Proteinase K can digest the cargo. So now, importantly, if we incubate together the plasma membrane sheet with the EVs, but if we do not trigger acidification, the cargo is not digested by the Proteinase K. But when we acidify the buffer, so now we can see digestion, suggesting that in that condition, there is content release. And therefore, our conclusion here is that acidification triggers the content release of the EVs when they are incubated with target plasma membrane sheet, and everything is quantified here. So we perform a kinetics, and what you can see here is that the reaction is relatively efficient, because after 10 minutes of incubation, you can see that half of the cargo is released in the buffer, and the process is complete after 1 hour. So the next step, so now that we knew that the acidification was required, which suggests that it's probably the indirect pathway, we wanted to test if proteins are important for that process. And we wanted to test the requirement of protein at the surface of the EVs or at the surface of the target plasma membrane sheet. So to do that, we did indeed a pre-treatment with Proteinase K to completely shave either the donor EVs or the target membrane. And what you can see here is that when we do that pre-treatment, we abolish the content release of the cargo. So what you see here is what I showed before, is content release when we acidify the buffer. So now, even if we acidify the buffer, if we do pre-treatment either on the donor EV or on the target membrane separately, we block the content release. And that suggests that the protein at the surface of the donor EVs or the target membrane are important for the content release. And so the simplest interpretation of the work is that content delivery is mediated by a fusion process that remains to be fully demonstrated. And that fusion process is dependent of acidification and a presence of protein at the surface of the donor membrane and the acceptor membrane. So it would be a protein-dependent fusion mechanism. So that was very satisfying, but what we wanted to do was to recapitulate our discovery and confirm our result within the cell and live cells. So for that, we developed a new cell-based assay to really characterize and, more importantly, quantify the process within living cells. And again, you can find most of the details and more information in the recently published articles. So for that, we developed a new assay. And again, we focus on the HSP70, but this time we tag the cargo with a very sensitive form of luciferase that's called nanoluciferase. That has a very high signal-to-noise ratio. And as a control, we use a membrane cargo, which is a nanoluciferase CD63. So again, we generate donor cells, and you can see here the activity of nanoluciferase within the gel. So here you have the donor cells that express the nanoluc, so there is no cleavage. It's expression of the protein that it's complete and not degraded. And here we have the control protein CD63. So again, we did isolation of the vesicles through ultracentrifugation. We also performed flotation assay to ensure that we were dealing truly with vesicles. So very briefly, we loaded the 100G pellet in the bottom of the tube, and then we loaded 3 layers of sucrose. And the extracellular vesicles are expected to float in the 30% sucrose. So we collected that fraction, and then we tested again for the proteinase protection to ensure that our cargo was truly inside. And as you can see here, when we were dealing with the middle fraction, so if we treat only with Proteinase K, most of the cargo is protected. And again, if we treat the sample with Triton to permeabilize the vesicles, so then the cargo is digested by the Proteinase K. Yes, that is again a confirmation then the cargo is inside the vesicle and appropriate for a content delivery within live cells. We also perform a full characterization of the isolated EVs. And as you can see here, the EVs were positive for classical marker and negative for ER marker. So with that, we validated qualitatively the EVs that we isolated, and so then we could perform the uptake assays with live cells. So here, what we did first was to incubate the isolated EVs that contain the HSP70 tag with luciferase, and we fed acceptor cells that were originally unlabeled. And what we call here an EV uptake assay is really the monitoring of luciferase activity that remain associated with acceptor cells. So we have three cases, right? So the EVs could be only docked at the surface, or internalize, or release their content inside. So at that stage, we cannot differentiate and distinguish the different steps. We only measure the activities of luciferase, and we call that the uptake. So we perform those response. And what we can see here is we couldn't reach the saturation of the signal even at very high doses of EVs. Just to give you some motif of comparison, for instance, it's well known that if you are investigated LDL uptake and the binding to the receptor, so you will reach saturation already at 10 micrograms of LDL per milliliter. So here we cannot reach that kind of saturation. So we also perform a kinetics, and what you can see is there is an increase over time. We could not really reach a saturation here. And what we estimated is at 1 hour, the spontaneous rate is about 1%. So importantly, what we did also was to test the requirement of the temperature. And we use a well-established assay in which we compare the uptake and the binding at 4 degrees versus 37 degrees. So please note that at 4 degrees, you abolish your internalization because you block endocytosis, but you do not prevent protein interaction at the cell surface if they occur. So for instance, many viruses, or again, the LDL receptor, if you incubate LDL particles with the cells, so you will see a binding at 4 degrees. So this is not the case here with the cell model that we use. As you can see here at 4 degrees, we barely see any activities associated with the cells. So when we put all together this data, what we show here is that the uptake of EVs is a very low yield process. So again, we estimated that it's a 1% spontaneous rate at 1 hour. And we believe that it's not occurring through binding to a specific receptor because there is no activity at 4 degrees. So we believe that it's a fluid phase uptake, that it's not really specific and not really efficient. But we could measure it because we were sensitive enough, so we decided to go further for the characterization. So first, we wanted to ensure that the vesicles were indeed internalized, and we did colocalization study to investigate the fate of the vesicles and the cargo. So here we use HSP70 tag with GFP, and we perform a confocal microscopic colocalization. So as you can see here, once incubated on the acceptor cells, the GFP tag EVs would colocalize either with RAB5 marker for early endosome. You have one example here. But we could also find the GFP positive EVs colocalizing with LAMP1 marker for a lysosome, suggesting that the EVs and their cargo will follow basically the maturation of the endosome. And we did quantification of that. And what you can see here is that roughly half of the vesicle were localized within the RAB5, so early endosome, and the other half were localizing with late endosome or lysosome. That's confirmed what was already known and published several times in the literature. So nothing was very new here, but it was important to demonstrate that the EVs were truly internalized. And again, that suggests that the first step is to get into the endosomes. So then, of course, what we really wanted to test was content delivery efficiency. So for that, we use the uptake assays that I described below, but we added an additional step, which is cell fractionation. And we use that to mechanically disrupt the cell and separate the membrane fraction from the cytosol fraction. The rationale here is that if the cargo and the EVs are internalized and remain associated within the endosomal compartment of the lysosome. So then they should be associated with the membrane fraction when we do the mechanical disruption. To the contrary, if cytosolic delivery occurs through fusion, for instance, so at least a portion of the luciferase should be associated with the cytosolic fraction of the acceptor cells. So the goal here was truly to feed the cell with the luciferase positive EVs, break down the cell, separate the membrane and the cytosolic fraction, and truly quantify the amount of cargo that could be delivered inside the cells. So first of all, what we did is to validate our protocol. And so Emeline Bonsergent performed a mechanical disruption, so without any detergent. And what you can see here is a Western blot showing that when we process the cell for mechanical disruption, as expected, most of the HSP70 that is known to be a cytosolic protein is found in the cytosol. And so here you have the percentage of cytosolic activities or presence of the protein is the cytosolic fraction. So you can see that more than 90% of the HSP70 is found in the cytosolic fraction. So to the contrary, CD63, a membrane protein, is exclusively found in the membrane fraction, and there is almost nothing or traces in the cytosol fraction. We estimated that the background is 15%. So we can do that on the donor cell with Western blot as a control, but of course we can use our nanoluciferase CD63 or nanoluciferase HSP70 expressing cells, and the results are very similar. So 85% of the HSP70 will be exclusively in the cytosol, whereas the CD63 is mostly in the membrane fraction. So now that we validated our protocol, we were confident that we could test the efficiency of the EV content release. So for that, again, we fed the acceptor cells with EVs that contain either HSP70 or CD63. So again, because it's a membrane, so CD63 is expected to be as a negative control, always associated with a membrane, whereas HSP70 could be putatively released in the cytosol. So when we measure the cytosolic release, what you can see here is that roughly 30% of the HSP70 that was originally contained in the EVs can be now found in the cytosolic fraction, suggesting that up to 30% of the uptaken EVs can be released in the cytosol, perhaps through fusion with the endosomal membrane. So what I want to point out here is if we compare now the delivery process from the internalization process, so the limiting step is the internalization step. So I told you before it was about 1% per hour, so here it's 30%, which is more consistent with many membrane trafficking process. So we then wanted to characterize and understand what were the requirements for the content delivery within live cells. And so for that, what we did is we treated the cell with bafilomycin, a drug that is known to block maturation and acidification of the endosome. And with that, we could test our hypothesis and test the requirement of the acidification. So as you can see here, when we only measure the uptake, so again, the total amount of nanoluciferase associated with the acceptor cells after feeding with EVs, so there is no changes. But very importantly, when we determine the content delivery and when we measure the percentage of cytosolic release, you can see that there is a massive and very dramatic inhibition of the content release when we block acidification of the endosome. And of course, we also confirmed that when cells were treated at 4 degrees, we block both the uptake and the content delivery. So that key experiment really shows that the content delivery, that was about 30%, is pH dependent, as we demonstrated previously with the cell-free assay. So that suggests that something is happening in the endosome, and we believe it's fusion. So to test that hypothesis, we focus on a family of proteins that is called IFITEMs for interferon-induced transmembrane protein. And these families of protein is well known to block the entry and the delivery of many viruses. So we decided to use an overexpression strategy. So in our assay, we know overexpress either IFITM1 or either IFITM3 on acceptor cells. And as you can see here, IFITM's protein decorates the surface of the acceptor cells, but also the endosomal compartments. And the main difference between IFITM1 and 3 is that probably IFITM3 is more abundant within the intracellular compartment. So when we now perform the uptake assay and the content delivery assay within cells that were either the control cells or cells that were overexpressing IFITM1 or IFITM3, what you can see here is that presence of IFITM dramatically reduce the content delivery of the vesicles. Perhaps because they block also the fusion with the endosomal membrane. Very importantly, we confirm that with our cell-free assay. So we use what I described in the first part of the talk again. So we had isolated cargo, isolated vesicles that contain the cargo, and we incubated with the isolated plasma membrane sheets that in that condition contain nothing or either IFITM1 and IFITM3. So here is what I showed before. When we do acidification, normally there is a massive release of the cargo. And when we have IFITM1 or IFITM3 in the target plasma membrane sheet, we completely abolished the content release. We believe it's because we block the fusion reaction. So that's true for IFITM1 or IFITM3. So one prediction, if we block the fusion, is that one would expect to see accumulation of HSP70 cargo trapped in an endosome, and that would be decorated with IFITM. And that is exactly what we observed. So here you have one example that perfectly illustrates that phenotype. So what you can see here, it's probably an endosome that contain IFITM1 in its membrane. And you can see here that probably several vesicles containing the GFP cargo are trapped and cannot deliver their cargo even after a long time incubation. And that can be quantified here. So we have more than 90% colocalization between IFITM protein and EVs. So that truly suggests that the EVs can be internalized, but they cannot deliver anymore their contents because we block fusion with IFITM, exactly as it has been described for viruses. So with that, I would like to conclude and summarize our most recent result. And here is the simplest working model that we would like to validate and we are working on. So we believe that the extracellular vesicles will be non-specifically internalized within the cell. So then we believe that acidification is going to trigger the content release. And we believe that that content release is dependent of proteins that are expressed at the surface of the vesicle and surface of endosome. And we believe that the content delivery occurs through membrane mechanisms that resemble fusion mechanisms that remain to be formally demonstrated. And that's our future goal. So now we are developing new assays to demonstrate formally that fusion truly occurs. And we now expect to identify the core machinery that governs that reaction. So with that, I would like to thank you, and I'm happy to take any question.
Okay. Let's stop it. Yeah, thank you so much, Greg. It's wonderful presentations and very clearly describe what the mechanisms and the effect that's involved in the cargo release. So I saw that what your data showing that there seems to be a lot of EV cargo is being associated with the lysosome. So would that sort of suggest that the cargo is being degrading, most of the EV cargo, or do you have any other hypothesis about what's happening?
So initially, what we wanted to do is to have a complete map of the fate of the content of the EVs. And so the initial goal was to measure what could be released which is what we did. And then we wanted to accurately also measure what would be degraded and what would be recycled. But we couldn't really measure that because it was too difficult to do that accurately. So I believe that when you see the 50% colocalization with LAMP1, does not mean that they're going to be for full degradation. Part of them will be acidic, like late endosome, if you wish, where the content release would occur. So probably, a significant portion of the LAMP1 positive structure could be efficient late endosome or early lysosome that will do the job for the content release. So to answer more precisely to your question, I don't think that 50% of the cargo would be destroyed. It's only a portion of it.
And also in the light of the EV heterogeneity, that will sort of make it very complex. What do you think about it?
Well, so what we are lacking now is to have, so the limitation of our assay is we need a soluble protein to test the content delivery. And we couldn't find many, cytosolic markers that would be, actually, we couldn't find any that would be specifically enclosed in one well-identified subtype. So we have all the tools. What is important now is to have one marker that would be specific for a family. So we have some clue and there are some candidates, but nothing that is formally demonstrated. So that's why initially, because we have very poor understanding of the general process, we focus on the generic marker because we know that HSP70 is probably found in many types. And the bet is like if there is truly a machinery for fusion, so it's probably a family of proteins. So my vision is if we do the analogy with SNRS proteins that are involved in intracellular compartment fusion, so it's a family of proteins, right? So each compartment will have a specific snare pair. So it could be the same for the fusion machinery for the EP. So each subtype will have a dedicated isoform, if you wish. So that could be a possibility.
Yes, fascinating. George Grau. Hi, George. Would you ask your questions, please? You're still muted. Yeah, okay.
Hi, Carolina. Thanks, Gregory. Excellent talk, really. I'm just wondering if you tried to block this fusion phenomenon using antibodies to surface molecules such as cell adhesion molecules or other surface elements.
So we didn't try with that kind of antibody. What we try is an antibody against LBPA. Also, we couldn't find any effects. But I have to be cautious with that because we didn't control the fact that the antibody was truly inhibitory. So it could just it's possible that it would just label LBPA without inhibiting the function.
Or antibodies to surface molecules of the EVs themselves. Do you think there would be a chance?
So we did some experiment in which we have cells that were knocked down for CD63 or CD9, for instance, and we couldn't see major effects.
Okay, thanks.
So I don't think that in our case, CD9 or CD63, I don't think that they are required, but we need a further experiment to fully validate that.
Thanks very much. Thank you. Great.
Okay, Sharif. Thanks so much, George.
I have a question about if you have any idea or tried something like trying to modify if we modify the protein content of the vesicles, or we loaded some specific protein that would direct the vesicles or allow them only to fuse with specific types of cells, and so we can use them as for targeting for certain type of cells like cancer cells or a specific type of cancer, something like that.
So that's a good idea, and we had the same. So what we are doing now in the lab is we are forcing interaction between the EVs and target cells. So with either an antibody and an antigen or purely artificial pair of binders, if you wish. So then we will have complete control. So the idea is to control the binding without touching the fusion. Now what we have also in our hands is a fusion protein So we can force and improve the fusion. So one of the examples is VSVG, for instance. So we can put the VSVG at the surface of the EV. We can also put other fusion protein that I cannot share right now because it's not finalized. But so the idea at the end is to have absolute control on the docking stage or on the fusion stage, and then be able to combine both.
Other questions maybe? No? Okay, so I think we probably closed earlier. Thanks so much, George, Greg, for your wonderful talk and presentation. So for next week, just quickly.
Sorry,
sorry, yeah.
sorry to interrupt, but I think in the chat there are other
questions.
Is there?
Oh,
perhaps,
yeah.
Oh, I think there is a last one.
Out. Go ahead,
if
Esther.
Okay, I can take that one.
Yeah, definitely.
Yeah, probably an obvious question, Gregory. Thanks a lot for your talk. It was very interesting. But did you try different combinations, and especially for the efficiency of this delivery process?
Yeah, so we tried several, and so what we did is we didn't change the donor cells, so it was always HeLa cells, but then we changed the acceptor cells, and so we tried mostly monocytes, dendritic cell model, or even HEK, and we couldn't see significant increase or differences even with monocytes. So I mentioned the 1%, so max will have 4%, something like that, but nothing very dramatically, no dramatic increase.
Yeah.
So that was
what would that mean to you biologically?
Well, it means that none of the cells that we tested are equipped with a bona fide receptor. That's my interpretation. If you don't have dramatic increase of the uptake, it means that the limiting step is the increase. And even if you specialize cells that are supposed to do more engulfment of the vesicles, if you don't have a bona fide receptor, it's still not a very efficient process. Also, what I want to mention, Esther, is so we try also interspecies. So one of our models was mouse acceptor cells, and we could see the same low efficiency for the uptake. That's consistent with several studies before. So in our hands, in our model, we don't see, species dependency. And to me, also signs go in the same direction. That, there is no highly specific mechanism for the uptake, at least in the cell type that we use. I mean, what would be interesting is to have a pair of donor and acceptors that is truly dedicated, and that would be physiologically relevant. I think that's what we need in the field. Something that is well defined and in which the efficiency is higher.
Yeah, but do you think there is a chance that the receptor-mediated uptake would result in a less efficient delivery than the process you're studying now?
Well, I think it would be more. If you increase the docking, everything that's downstream would be more efficient. To have efficient fusion, you need efficient docking first. So if you improve the docking, the fusion will be higher.